So what I wanted to talk about today is a little bit of a subject that a lot of people, rightfully so, ask a lot about. And sometimes I would say that I hedge a little bit and not give particularly clear and direct answers. And a lot of the times it's because I don't have a very clear and direct answer. But the question is, which is a sense has nothing to do with virology. But if it isn't viruses, then why do people get sick? And I've, of course, done a number of talks on that, and I have a whole model of what's real in tissues and cells, and the whole idea about that we're basically made of organized water, and we have nuclei that participate in the downloading of information from our thoughts and feelings in the sun and the moon and Venus and plants and dogs and cats and everything else. We download that into our water and create life out of that. And when something goes wrong in that system, either poisons in the water or something destructures or decreases the coherence of the water, or you get the wrong kind of signals like abnormal electromagnetic fields, or not enough love or support in your life, or thinking delusional thoughts, and all, etc., that essentially creates interference or distortion in the system. The body tries to remedy that, and that's what we call disease. So I've been through that and given a lot of examples, but today I want to show a clip, or actually extensive clips, from somebody who I don't know anything about. I don't know his background. I was just sent this talk, I guess, that he gave in front of a group of scientists and virologists, talking about why people get sick. And I thought there was a number of things in there that actually are useful. They're not particularly new. It's not something that none of us have said before, but I thought they were presented in a fairly clear and succinct way. And so it will help people get a better handle on the question. So we know it isn't viruses, so why do we get syphilis and polio and the rest of it? The other thing that I want to say about this clip is, unfortunately, as far as I can tell, the speaker, his name is, I think, Sebastian Powell, of course, does homage to the virus king. And I'm not sure why, because basically his whole talk is describing that there is no viruses, and every symptom, every illness is better explained in some other way. But he still, again, pays homage to the virus king. And I'm not sure why that is. But he also gets into the difference between exosomes and viruses. And here I do want to point out that, in my opinion, and maybe I'm misinterpreting what he's saying, but there is a fundamental distinction between what are being called exosomes and what are being called viruses. The distinction is a exosome is defined as a piece of the tissue or cell that, because of certain processes going on in the tissue or cell, particularly having to do with illness, in other words, something makes the tissue or cell sick, and it could be any number of things, the cell breaks down, or the tissue breaks down, and as it breaks down, it creates these particles which have the identical size and shape and composition as to what are called viruses. They are, however, they do not replicate, unlike what we're told about imaginary viruses, and they are not infectious. They do not cause disease, and they don't code for any proteins as far as we know. So those are major distinctions between what we are calling, or what the virologists call, the definition of a virus, which is a replication competent protein coded genetic material, DNA or RNA, in the inside infectious particle, which causes disease by replicating and essentially exploding or destroying the cell as it exits the cell. So there is a fundamental distinction. And as I've pointed out a number of times, I'm actually not right now convinced that there is such a thing as an exosome in living tissue or living systems like humans, that what we're looking at is basically just cellular debris that, when prepared for electron microscopy, has the same shape and morphology as what we used to say were, quote, viruses. Now I'm going to talk a little bit about out of both sides of my mouth. So on the one hand, I'm not convinced that there is such a thing in us as we're living as an exosome. I think we're just talking about debris, which has no function and no role in biology. So that's what we call an exosome. And there, everything that has been looked at as far in virology to say we see this particle does not represent an infectious replication competent particle. It just represents debris coming from the inevitable breakdown of the tissues and cells. And again, under an electron microscope, it looks like what we used to call a virus. So hopefully that's clear. Now, an astute listener and reader of mine, I guess, sent me an interesting article, and I'm going to have to explain why I'm even showing this. And the reason is, so this is a paper, you can see the published in Current Protocols in Cell Biology, volume 88. I think it was published this year, or maybe in 2020, Wiley periodicals here. Now, the purpose of this paper was to elucidate the technique that they used to purify and isolate exosomes in the blood of people, so, or mammals maybe. So exosomes are 50 to 150 nanometer diameter extracellular vesicles secreted by all mammalian cells except mature red blood cells, and they say contribute to diverse physiological or pathological functions with the body. I'm not so sure about that. And then they talk about the difficulty that people have had with purifying them. And then they present protocols for exosome isolation using the two most commonly used methods, ultracentrifugation and precipitation. And then they go on to demonstrate how, using these techniques, they were able to get pure exosomes. So why am I showing this? Only to say that they were able to purify and isolate particles that are not viruses, but have the exact same size, shape, morphology as what are thought to be viruses, 50 to 150 nanometers in diameter. And so it demonstrates the, I guess you could say, lie that there is no way to purify such a thing or isolate such a thing, the size and complexity of a virus, directly from the blood or tissues of animals or humans. Because this is actually what you can find, are these exosomes, which are identical size and shape. Now, to be clear, again, they are not viruses, and in fact, they are probably actually artifacts. But you can purify these artifacts because what you see on the electron microscope will spin out at a certain band. And then when you stain them and prepare them for an electron microscope, you see pure exosomes in this band. Granted, they're not reflective of what was there in the living person. That's what I mean by artifact. But still, you can get a pure product that is the same size and complexity as a virus. The importance of this is to demonstrate that it's not true that we couldn't find something like a virus directly from patient samples, which is why when you compare this with Christine Massey's work, you can see that it is a total misconception to say she is asking for information that isn't possible, because clearly it is possible. If you just change the word from virus to exosomes, you would easily be able to isolate them and separate them and purify them directly from patient samples. No need for any cell culture. And again, the only reason they have not been able to demonstrate that is very simple, because they don't exist. They're not there. There are no replication competent infectious particles called viruses. There is only cell debris, which is easily isolated, purified, studied, characterized. It has no function. It doesn't even look like that probably in real life, but the techniques are there to get it. Okay, so with that, let's go on to this clip, and I'm going to play a number of them, and then make comments along the way. I got the wrong one. Okay. I don't think I can make this bigger. Maybe I can here. Yeah, I can. Okay, first clip about polio.
Said to be a distinctive symptom of a poliovirus infection. According to the CDC, only 25% of those who are infected will develop symptoms, which are said to be flu-like, sore throat, nausea, headache, etc. Of these, between 1 to 5 in 100 will develop meningitis, whilst 1 in 200 or 1 in 2,000 will develop paralysis depending on the virus type. The CDC also states that, quote, polio is defined as the paralytic disease, so only people with the paralytic infection are considered to have the disease. They further assert that the virus has been, quote, around since ancient times. Evidence for this statement is provided in the form of this Egyptian tomb painting depicting an individual with a, quote, withered leg, a condition known as post-polio syndrome. However, it turns out that a number of microorganisms, conditions, and substances are known to cause the exact same symptoms.
Yeah, I just want to point out the first thing here. So this is a common thing that happens in medicine. The CDC or some other institution will assert that a non-specific finding like paralysis or atrophy of a leg is pathognomonic, means it is the absolute hallmark of polio, and then only to find out that's not the case, and to use that as a as proof that this disease called polio existed in Egypt because of a withered leg on a stone, on a carving, is pure nonsense.
Include non-polio enteroviruses such as Coxsackie virus, other viruses such as varicella zoster virus, rabies, and SARS-CoV-2, autoimmune diseases such as Guillain-Barré syndrome, genetic diseases such as Charcot-Marie-Tooth disease, bacterial infections such as tuberculosis.
Again, I want people to ignore the homage to the germ theory here. None of these viruses exist or cause this, but what you can interpret this as, there are a lot of other things besides what are called polio that cause people to have atrophied and withered legs.
Parasite infections such as Plasmodium, which on occasion is said to cause cerebral malaria, and various poisons such as lead and arsenic, and even nutritional deficiencies such as scurvy. A report from 1993 details the case of a 4-year-old boy who was initially diagnosed with, quote, acute paralytic poliomyelitis. His diagnosis was subsequently revised to that of scurvy, which suggests that two diseases present themselves clinically in ways that are indistinguishable. There are many other such examples. To illustrate these similarities, on the left is a polio victim, followed by a beriberi victim, which is said to be caused by a vitamin B1 deficiency, and finally a victim of the English beer poisoning of 1900, where it was reported that some 6,000 people were poisoned by arsenic-tainted beer. It is therefore unclear on what grounds the CDC feels it is appropriate to assert that the Egyptian tomb painting depicts an individual whose condition developed as a result.
So again, this is a common thing we experience. You have a set of symptoms. The same thing is happening now. The authorities say that this is a hallmark of such and such a disease, when it turns out that it's by no means a hallmark of any specific disease. And he calls them all kinds of viruses and bacteria. But as you can see, they're all also hallmarks of various kinds of starvation and poisoning. So let's move on to something to do with syphilis.
The same pattern appears to apply to diseases said to cause specific rashes or skin lesions. On the left, we have a picture of a smallpox victim, and on the right, a picture of ex-Ukrainian President Viktor Yushchenko, who was the victim of dioxin poisoning in 2004. Physician Al-Razi, who was born during the 9th century in Persia, was apparently the first to describe the symptoms and signs of smallpox and measles. He is reported to have written that, quote, back pain was more severe in smallpox, while it might be slight or absent in measles. Nothing appears to have been said concerning the appearance of the rashes. Another example is syphilis. According to a paper published in 1902, prior to the 15th century, syphilis and smallpox were confused for one another. According to the CDC, quote, chickenpox was not reliably distinguished from smallpox until the end of the 19th century. It is unclear why so much time had to go by before people realized they were separate diseases, if that is indeed what they are.
So again, we have another example of people can't tell the difference between measles and smallpox and measles and chickenpox and measles and poisoning and measles and this, and you go on with, and we remember a video from Kate Sugak where if you want to say something is an epidemic, you say all these various symptoms are one disease called COVID or whatever, smallpox or anthrax or polio. And then when you want the epidemic or pandemic or whatever to end, you go back to separating them and saying, no, those aren't actually smallpox or polio. That's beriberi and scurvy and arsenic poisoning and all the rest. Very clever trick they use. So let's go on. By the way, I don't have the link for this, but people might want to take a look at this. It's got some interesting.
Unwell. For instance, a paper titled The Fever Paradox, published in 2020, notes that, quote, mounting evidence suggests that fever may in fact be a strategy the body uses to ramp up its defenses. This idea isn't new. In 1859, Florence Nightingale wrote in her book Notes on Nursing that, quote, all disease at some period or other of its course is more or less a reparative process. Infectious diseases are those said to be.
So again, and I like the Florence Nightingale quote, all diseases are some sort of poisoning or some sort of nutritional deficiency. They're not contagious. And the symptoms that we see are a body's attempt to repair the damage. Pure and simple, no virus needed, no bacteria needed. That's what's happening. Diseases are starvation, poison, injury, delusions. The symptoms are the body's attempt to repair the situation, and that's all there is to it. Okay, let's skip around a little bit here.
The researchers conclude their report by stating that, quote, it would appear that the monkeypox virus may be carried by some species of monkeys as a latent virus. Irradiation by decreasing host resistance may lead to the appearance of clinical disease. It would appear that it is inconceivable to them that the symptoms are in fact symptoms of poisoning. Ionizing radiation is of course contagious in the sense that people who have been exposed to radiation remain radioactive for a period of time. For instance, people who are treated with radioiodine.
So I didn't put the context here, but he's saying that when we see people who we claim have monkeypox and we say there's a virus, in some cases it has been the result of radiation exposure. And in this, now he's going on to make the case that to a certain extent, radiation exposure is contagious, or I think the better word is transmissible, because even with people who are given radioactive iodine for treating thyroid cancer inappropriately, I would say, they seem to be emitting radiation, which can have a negative effect on others, making it seem like something is being transmitted. Well, something is being transmitted, but certainly not any microorganism. It's just if you radiate somebody, they emit radiation, and this is a well-known fact in conventional medicine. That's why they say 16 days, avoid staying within 6 feet of others, no kissing or sexual contact, etc. So it's easy to understand that you can get symptoms, even possibly being around others, that is seemingly transmissible. It is sort of transmissible, but there's not anything contagious in the usual sense of it. Now, some may ask, is this even happening with vaccines? And I don't know that, but I guess it's possible. And I know I'm skipping around here, but
when we consider the idea of the so-called human virome. According to an article published by Cold Spring Harbor Laboratory on this topic, quote, 8% of our DNA consists of remnants of ancient viruses, and another 40% is made up of repetitive strings of genetic letters that is also thought to have a viral origin. So it sounds like we're mostly made up of these viruses, and perhaps this is where dormant viruses come from. Going back to the original monkeypox paper, two things.
So let me just say, this idea that we're made up of viruses is not true. It's the opposite. We have all these strings of genetic material, most of which has no function that we know of to code for proteins. And when the tissue breaks down, as it inevitably does, and worse when you're poisoned or sick or starving, then they get encapsulated in these little microvesicles, i.e. garbage, sometimes called exosomes. And those are inappropriately called viruses when in fact they don't replicate, they don't cause disease, and they're not infectious. So that's where the origin of that stuff came from. I'm not sure if he understands that, but there is no virome. There's just a bunch of genetic material that breaks down into particles.
Stand out. The particles that are allegedly the cause of the deleterious effects have not been isolated. And two, the particles are transmitted to the test subjects in a completely unnatural way that bears no resemblance to what is said to happen in the real world. This appears to be another common theme in this field of study, going all the way back to Koch and Pasteur. For example, in the case of anthrax, Robert Koch discovered that, quote, inoculating a mouse with blood from a sheep that had died of anthrax caused the mouse to die the following day. As we have previously discussed, bodily fluids like blood can contain any number of contaminants, including poison, and in this case it seems exceedingly likely given the widespread practice of using arsenic-based compounds such as Cooper's dip for the purpose of sheep dipping.
So here he's making the case of how illness can be transmitted, especially in animal studies. So you take an animal like this sheep, unfortunate sheep, who gets dipped in poison, and then their blood is contaminated with arsenic, and then you take the some of their blood and inject that into a test animal like a mouse, and the mouse gets sick, and you claim that you have transmitted some microorganism to the unfortunate mouse. In reality, what you've done is poisoned the sheep. Some of the poison remains in the blood, and then when you inject that blood containing that poison into the test animals like the mice, they, no surprise, also get sick, and no virus obviously needed in any of this process.
Paralysis. The emulsion was injected into the peritoneal cavity of the monkeys. On the subject of so-called infantile paralysis, it is interesting to note that a full-grown man, President Roosevelt, woke up the day after going for a swim in a lake near his summer home one day to find himself partially paralyzed. He was eventually diagnosed with infantile paralysis, or polio. In 1923, Thomas Rivers published studies on varicella, where he attempted to demonstrate the contagiosity of the varicella zoster virus, which previous studies had failed to do. Rivers writes, quote, Hess and Unger failed to produce varicella in normal children by inoculating them upon the mucous membranes of the nose and throat with vesicle lymph. Quote, Kling stated that to obtain positive results, at least 6 inoculations should be made, and that even then only 1 may be successful. These reports seem to indicate that the virus in the vesicles either is very dilute or has lost its virulence.
So this is a study going back to 1923 that the attempt to transmit chickenpox with direct exposure to the contents of chickenpox vesicles, even by the so-called father of early virology, Rivers, failed. They should have dropped it right then, but unfortunately they didn't.
This is curious given that the vesicles are said to be one of the routes of transmission of this virus. According to Rivers, these observations suggest the virus must be somewhere in the blood, and therefore that, quote, it might be possible to demonstrate the disease in animals by injecting them with patients' blood. He then proceeds to perform various experiments on rabbits using the blood of infected patients. Quote, blood was drawn from patients with chickenpox, usually during the first 24 hours after the appearance of the eruption. The blood was not citrated, and before clotting occurred was injected in 2 cc amounts into each testicle of normal rabbits. Quote, at the time of inoculation, the needle was moved about in the tissues to produce a certain amount of trauma. 4 days later, the testicles were removed, ground up thoroughly with sterile, chemically clean sand, and mixed with 10 cc of physiological salt solution. Then 1 cc of the emulsion was injected into each testicle of normal rabbits. This is just a small sample of what he did. Other experiments involved injecting the emulsified materials into the eyes of rabbits. I would recommend reading the paper in full if you have the stomach for it.
So again, very interesting. These early studies, they showed that if you injected unpurified material into the testicles of these poor rabbits, I don't know if I got this exactly right, and then you had to induce the trauma into the testicles, that might produce an irritation and a sort of kind of a symptoms in the testicles. And then you grind that up and mix it with sand and inject that into more testicles of more rabbits, and you might get symptoms for that. And all that proves is if you're a rabbit, you better stay away from medical experimenters, because It's not going to be good for you. It's not going to turn out well. And it has nothing to do with the transmission of any so-called virus.
What was done back then. So I asked a virologist on Twitter who argued this exact point for an example of a modern paper. He shared one published in 2012 where they attempted to transmit the vaccinia virus to mice. The methodology involved shaving and anesthetizing them, and then, quote, applying 1 × 107 platforming units of VV, vaccinia virus, to the dorsal skin and scratching 20 times with a bifurcated needle. The end result was this. I asked the virologist in question if he didn't think that the simple act of causing this sort of trauma might lead to the formation of these kind of lesions. In response, he said, I don't work with mice. Apparently, one needs to be an expert to use your critical faculties and common sense.
Right. Common sense would say scratching mice, the skin of mice on their sensitive belly with a bifurcated needle that's dipped in all kinds of toxic cell culture goop might not be good for the mice and might create some sort of lesion. And apparently you have to be a professional virologist who works with mice to get that, the irony of that statement. Okay, next.
Another example is that of malaria. In 2014, malaria vaccine researchers allowed monkeys to have plasmodium transmitted to them by infected mosquitoes, as opposed to injection. The study notes that the parasite was transmitted, however, no mention is made of any monkeys developing any symptoms. It is possible they simply didn't mention them, as this wasn't the purpose of the study, but it is interesting nonetheless. If the
Okay, I'll skip that one for a minute, but here, let's keep going.
particles that scientists refer to as virus are not in fact contagious or indeed the cause of disease, then what are they? In a study published in 2022, researchers in China found that, quote, circulating exosome levels may be novel biomarker for ecotoxic effects of water environment. In other words, they found more exosomes in fish exposed to various pollutants, including arsenic and mercury. Quote, the increased circulating exosome level of indigenous fish samples were consistent with the oxidative damage and genotoxic effects of the water environment. Other studies appear to show similar findings.
So, in other words, when the fish are swimming in a poisoned water, they make more of these breakdown products from their tissues, which are called exosomes and given certain properties which they don't deserve. Why that's a surprise to anybody, I don't know.
In a paper published in 2018, researchers found that there was a negative correlation between lung function in the case of, quote, progressive fibrosing interstitial lung disease of unknown etiology and levels of exosomes containing MIRR 1423P, leading the researchers to conclude that these exosomes were, quote, promising biomarkers for diagnosis and disease severity. Exosomes, or extracellular vesicles, are nano-sized particles that appear to bear an uncanny resemblance to viruses.
Actually, they are identical to what we are calling viruses. And as we've said many times, nobody with any kind of microscope or electron microscope can tell the difference. So there is no case to be made that these particles seen on electron microscopy are actually infectious, replication competent particles that cause disease as we think of as a virus.
On the left, we have cryo-electron microscopy photos of extracellular vesicles, and on the right are photos of coronaviruses.
Except, how does he know these are coronaviruses? And the answer is he doesn't. They are said to be.
Ultracentrifugation is one method that is commonly used to allegedly separate viruses from other contaminants. However, there appear to be problems with this technique. According to a paper published in the journal Viruses in 2020, quote, "Nowadays, it is an almost impossible mission to separate EVs, extracellular vesicles, and viruses because they are frequently co-pelleted due to their similar dimension." Another key difference between exosomes and viruses, according to an article published by Cell Guidance Systems, is how they are produced. Quote, "By definition, a virus can replicate by using host machinery. Exosomes do not replicate in recipient cells. Each cell has an innate ability to make its own exosomes. Thus, an exosome or other EV cannot be a virus because they cannot replicate." This sounds
exactly. They're just garbage. They can't replicate. They do not fit the definition of what we say a virus is, and they cannot cause disease. And that's all there is. Nobody has ever been able to say this is not an exosome, it's a virus. Not ever has that happened.
an awful lot like circular reasoning, and it is unclear how virologists know for certain that viruses replicate in the way they are said to.
Exactly. You cannot know. That's the reification fallacy. Since you cannot say this is a virus, how do they know they replicate?
Microscopy kills the cells, and therefore this process has never, as far as we are aware, been observed in real time.
Right. They've never seen this. You can only see this under an electron microscope, which means it's already not able to replicate. This is a theory, not a theory. It's a claim that needs to be discarded. Okay. Let's move on a little bit.
Called a cytopathic effect. Not only that, but exosomes also appear to contain viral RNA. In a paper published in 2021, researchers found that, quote, "Analysis of exosome content purified from critical and non-critical patients revealed the presence of SARS-CoV-2 RNA in the exosomal cargo."
And again, since they're identical and even have the same kind of genetic material, supposedly, then how can you possibly say this one is a, quote, virus or SARS-CoV-2 and this one is an exosome? You can't.
In fact, it would appear that exosomes play an important role in the infection of cells. A paper published in 2019 found that...
How does he know it's an infection if he can't prove there's a virus?
Quote, "Rabies virus infection increased the release of exosomes. The role of exosomes in virus infection process remains unclear and needs to be further investigated."
Or discarded, I would say. Let's move on.
For instance, mosquitoes are able to contribute to the transfer of toxins and therefore spread of disease. A number of studies have found that mosquitoes, beetles, ticks, and other insects have developed arsenic resistance, and that it will bioaccumulate in them, as it seems to in all living creatures. In a study published in 2018, researchers wrote that mosquito saliva involves more than 100 proteins. Some of these, such as anticoagulant AAPP, contain a number of sulfur-rich cysteine residues. This in turn suggests that mosquitoes exposed to arsenic when injecting their saliva may also be injecting some of the arsenic they've bioaccumulated. It would be interesting to devise an experiment to validate this, as we've not been able to find one.
So this is a very important point. I skipped over this a little bit, but the point he's making is, and this is especially interesting, I think, to people who say that Lyme disease is an infection, that since no spirochete has ever been found to create disease called Lyme disease in any humans or animals, as far as I know, any purified, isolated spirochete exposed to people doesn't cause disease, then you have to start wondering whether, if he's correct, which he suggests we need more research, that there are at least 100 or so other enzymes and proteins in the saliva of biting insects, and that these insects, like ticks, actually bioaccumulate toxins like arsenic in their saliva, and they may be injecting the arsenic that they bioaccumulated into you, thereby causing symptoms which are erroneously ascribed to a bacteria. And then when your body tries to eliminate this toxin through rashes and swelling and inflammation, because you or your doctor don't understand what's happening, you use antibiotics, suppress it, guaranteeing that now you're more toxic and that the symptoms will eventually reemerge. So that's a very interesting and coherent way of explaining everything that you actually experience with insect bites, tick bites, and all the rest of it.
Serious diseases that seem to regularly crop up swept through Cuba. This virus was once again found in samples taken from sick patients. Quote, all cerebrospinal fluid specimens from 20 Cuban patients tested were positive for IMV type 2, and 4 cerebrospinal fluid specimens from Cuban controls were negative for IMV. Microorganisms being markers for toxicity is a pattern we also find for both parasites and bacteria. A study published in 2007 found that, quote, helminth parasites are extremely sensitive early warning bioindicators, particularly in sensitive environments under threat but where pollution levels are presently low. They may also have a beneficial effect on the health of their hosts by acting as heavy metal filters. In a study published in 2017, researchers found that, quote, the concentrations of...
So let me just comment on that, which is, again, something I've been saying for a while. If you go back, you can see that helminth, which I guess is a kind of a parasite, are sensitive to pollution and toxic levels in the environment, and they have a beneficial effect on the health of their hosts by acting as heavy metal filters. Again, the sequence of events is exposure to heavy metals through a variety of means, including maybe insect bites or things sprayed in the air or injections of unfortunate so-called vaccines. And then you end up with a parasite growing in your tissues, which is a way to act as a heavy metal filter and to help you out. And unfortunately, the doctors and the patients don't understand this, so they go about killing the parasites, which is your first line of defense. And that, of course, makes the person sicker because now you've used a toxic parasite killing stuff, ivermectin, et cetera. And so you have the toxicity of the treatment, plus now you have heavy metals and other biotoxins which cannot be filtered out by your friends, the parasites. And so obviously you get worse and worse and worse. That's the problem with not understanding how disease works and what the cause of people's illness. So no matter how many times I hear from doctors, we don't need to know whether there's viruses or not. We don't need to know the cause. It's total nonsense. Okay.
Quote, Fleck demonstrated that molds growing on wallpapers that were painted with arsenic pigments such as Shields green and Schweinfurt or emerald green were able to produce a volatile arsenic compound. Given all of this, it would appear that there's quite a reasonable explanation for the never-ending plethora of seemingly disparate diseases and their causes, going right back to the so-called Victorian diseases that were running rampant at the time. And by this, I mean more reasonable than the apparently commonly held view that, as an article published by Imperial College put it, quote, nature is trying to kill you, but science has your back.
Right. This is something we talked about before, that it turns out, so why do similar children in this, or children in the same house or in the same school get sick at the same time with the same symptoms? There's obviously a lot of potential reasons, but one of them could be the arsenic in the paint or in the wallpaper. and something changes maybe in the weather or the humidity or in the cleaning products, and the arsenic gets volatilized, and all the children end up with rashes, which are indistinguishable from the rashes we call chickenpox or measles or many other names we give to the same thing. And it could all be a toxic exposure of a similar nature. Nothing is being transmitted, just a similar environment. Okay, just a few more. There was a couple interesting ones.
Predisposition to autoimmune disease. To take just one example, researchers recently found that in the case of cystic fibrosis, quote, CF, cystic fibrosis, is caused by a genetic defect in the CFTR protein, which regulates the flow of chloride in and out of cells. Recent studies in cell culture show that arsenic causes the CFTR protein to break down. There are similar findings in relation to, for instance, Charcot-Marie-Tooth disease, which is also said to be a genetic condition.
So here we're talking about, I've said this a number of times, people are told all the time you have a genetic condition, and the genetic conditions seem to be increasing in leaps and bounds, even though there's no evidence that our, quote, genetics is changing at all, except I guess if we're trying to change it. I mean, the genetics is probably changing due to environmental toxins, but what's really happening here is people are being poisoned, and their symptoms, their actual illness, is from the poisoning. In this case, he's talking about arsenic and lead. And coincidentally, not so much, but you see other things break down, including the genetic material in the tissues and cells also breaks down, which has nothing to do with the disease. It's just yet another chemical found in us, which obviously breaks down when it's poisoned. So those two things have no causal relationship. The change in the so-called genetics and even the formation of the proteins or the breakdown of the tissues, i.e. the disease, they're just both manifestations of a similar poisoning.
This is worth discussing. Given that the particles called viruses are never properly isolated and purified, it is unclear how exactly these genes.
I just want to point out that, as he says, we all know that none of these viruses have been isolated or purified. So we already know that.
Genomes are sequenced and what it is they actually represent. According to the NIH, PCR is a
Okay, we all know that already. And the final one, which I thought was interesting.
Malaria actually provides one of the best examples for how differences in testing protocols distract people with endless lists of diseases and alleged pathogens and ignore the common denominator, which appears to be mass poisoning. Malaria is said to have been eradicated from most northern African countries, including Egypt. The situation, however, is quite different in neighbouring Sudan. According to the UK's National Health Service, the risk of infection is considered low in the northern part of the country and high in the southern part. One might expect some of the mosquitoes present in the northern part to eventually find their way over the Egyptian border. However, this doesn't seem to happen very often, for reasons that are unclear, even though researchers in Tanzania found in 2019 that, quote, mosquitoes can travel almost 300 kilometers in a single night. Although there is no malaria in Egypt, a study published in 2009 found that typhoid fever is a problem in most North African countries. And of course, the symptoms of typhoid fever are virtually identical to those of malaria, yellow fever, dengue fever, West Nile virus, St. Louis virus, Rift Valley virus, Lassa fever, Crimean Congo fever, Q fever, Zika, etc.
again, we have this similarity of diseases. They all have the same symptoms, more or less. And if you want an epidemic, you call them all one. And if you want to have many different viruses to give many different injections or treatments, you say they're all different and based on the pieces of genetic material that are being broken down. And I thought it was very informative to point out that when you actually look at the incidence of malaria supposedly transmitted by this mosquito, even though mosquitoes can apparently fly 300 kilometers in a single night, in one country you have no malaria, and in the other country you have some, and then in a different part of the country you have a lot, and the mosquitoes are freely flying from one place to another. But as he goes on to point out, the exposure to different biotoxins, particularly arsenic, is much different in these different places. And that would give a very clear and cogent explanation, whereas the flying mosquitoes carrying a bacteria explains nothing. So hopefully that was a good way to take a look at another way of looking at disease. And I think this fellow did a good job in explaining and elucidating that the virus story really needs to go, and we need to start studying actually why people get sick. And as I've said, we should start with the 4 biggies, which are people fall off their horse, they're starving for all kinds of things, they're poisoned. In his case, he talks a lot about arsenic and lead arsenic and Paris green and all the rest of it. So that's one example out of hundreds probably. And then finally, the big one is people are delusional, thinking in their mind somehow that these invisible things are out to get them. So I hope everybody enjoyed that, and we will again try to do our big announcement next week on Wednesday. I hope everybody has a good week, and I will see you next week.